Dear colleagues, ladies and gentlemen, my name is Christian Stevens and I'm the coordinator of the International Thematic Network on Green Chemistry of Ghent University and head of the SynBioC Research Group. The SynBioC Research Group is part of the Department of Green Chemistry and Technology, and I will present today some of our work on the development of natural product analogues and anti-cancer research. The SynBioC Research Group is composed of 4 professors, 4 postdocs, about 25 PhD students, 4 ATP staff, and about 20 master students. Our mission is the development of sustainable synthetic methodology for the synthesis of heterocyclic compounds with potential applications in the agrochemical and the medicinal domain. The 3 important research lines within my research group are the chemical modification of renewable resources related to the transition to a circular bioeconomy, the use of continuous flow chemistry using micro and mesoreactors for process intensification, and the reduction of the footprint during the upscaling of chemical reactions. The 3rd research line, of which I will be talking today, is the development of synthetic methodology for azo-heterocyclic compounds with the aim to create active compounds interacting with biochemical pathways. The applications can range from antibiotic applications over compounds with analgesic or antidepressive activity or compounds with anti-invasive properties. Today, I will briefly discuss 2 projects in the field of anti-cancer research. Globally, 1 out of 6 mortalities is caused by cancer. Of these cancer casualties, 90% is due to metastasis of the tumor and not because of the uncontrolled growth of the primary tumor. The metastatic process starts with invasion, which is the first and fundamental step of the cascade, making the inhibition of invasion very interesting from a therapeutic point of view. A few drugs are being developed in industry. However, until now, no efficient anti-invasive drug is available in the clinic. Our project is dealing primarily with breast cancer research, but in principle, the mechanism of metastasis is universal for solid tumors. In the next slide, the invasion process is depicted. The primary tumor splits off cells that migrate to a blood vessel by breaching the basement membrane and the cell matrix. The tumor cells then travel through the body to find a distant organ that is suited to receive the tumor cell and where a secondary tumor can start to grow. This process is repeated constantly and leads to showers of cancer cells traveling through the body, which can lead to an exponential formation of secondary tumors. The goal of our project is to develop inhibitors of this invasion process. The Laboratory of Oncology at Ghent University Hospital has been screening for many years hundreds of natural plant compounds in the chick heart assay. In this assay, healthy chick heart embryo cells are cultured and are then confronted with breast cancer tumor cells. If no active compounds are added, the chick heart cells get invaded soon and are transformed into tumor tissue after 7 days. When the screening compounds are evaluated, this process can be slowed down or even completely stopped when an active compound is added. On the left side of the slide, the tissue is shown in which the chick heart cells are completely transformed into tumor tissue. On the right side, it is obvious that no invasion occurred and that the surrounding breast cancer MCF7-6 cells are not able to invade the chick heart cell aggregates. It means that the added compound shows anti-invasive behavior at the tested concentration of 1 micromolar. From the initial screening of the plant compounds, it became evident that there was a certain structural pattern common to some of the active compounds. The chalcone structures shown here was present in several active natural compounds. When we started the synthesis project, we were convinced that by changing the substitution pattern on the aryl rings and the double bond, more active compounds could be developed. Since there is no evolution pressure on plants to synthesize compounds which are active against breast cancer, it makes sense that plants did not develop the optimal substitution pattern. Indeed, it was possible to obtain 3 compounds with 100 times higher activity. The most active compound kept its activity up to 10 nanomolar. In vivo experiments on mice showed there was a more than 40% reduction of invasion in kidneys and lymph nodes for the compound C16. The compounds could be easily synthesized by aldol condensation of the substituted acetophenones with functionalized aromatic aldehydes. Without going into detail, we could prove that compounds work through a novel mode of action. The compounds were also proven to have nanomolar potency in orthogonal invasion assays against ovarian carcinoma cells and BLM melanoma cells. The ADME and PK studies of the compounds are favorable and no red flags were detected related to toxicity issues. The project now continues and became a real multidisciplinary project with departments of several faculties in order to discover the exact mechanism of the chalcones. It is the intention to create a spin-off company in Q4 of 2022 and further develop one of the compounds to a marketable drug. Of course, we realize that there is still a long way ahead. However, we want to study this new mode of action in its full details since we believe we have a first-in-class anti-invasive compound. A second topic we have been working on is the synthesis of analogues of blebbistatin. Blebbistatin is a known inhibitor of non-muscle myosin II. Myosins are proteins that are responsible for all kinds of movement. They are actin-based molecular motors of the cell that convert the energy released by ATP hydrolysis into force and movement in an ATPase cycle. Myosin has been identified as an important regulator in motility-based diseases such as viral and bacterial infections and invasion in malignant tumors. The central role of myosin II in these diseases was revealed with the aid of the small molecule research tool Blebbistatin, which slows down the movement. In this little video, the mechanism is explained how myosin is creating the movement over the actin fibers.
With calcium elevated around the myofibrils, myosin binding sites on actin are exposed, allowing interaction of the myosin cross bridges with actin. Release of the myosin cross bridge from actin requires binding of ATP. After ATP binds, it is split to ADP and inorganic phosphate. The energy derived from this reaction is utilized to cock the cross bridge in preparation for the power stroke. The power stroke is initiated when the myosin cross bridge binds to actin. The cycle begins anew when another ATP binds to myosin. At rest, most cross bridges are in the cocked configuration, prepared to interact with actin when the tropomyosin-troponin complex is moved away from myosin binding sites on actin by calcium ions.
The characteristics of blebbistatin are not ideal to be used in cells, primarily because of its very low solubility and its fluorescent properties, which compromises certain cellular techniques. If you want to see certain fluorescent labeled compounds in the cell, such as the biking people in the dark, for example, the fluorescence cannot be troubled by a high background fluorescence of blebbistatin, as can be seen on the picture on the right. Therefore, blebbistatin analogues are needed with good myosin inhibiting properties and with better physicochemical properties which are listed here. But above all, the light stability of the compounds need to be improved as well as the selectivity, and this all combined with a low toxicity. The goal of our research was to study the synthesis of blebbistatin derivatives. The idea was to keep the blebbistatin scaffold, of course, but to make small alterations to different parts of the molecule. We have modified 3 rings of blebbistatin in order to find out which parts were crucial for activity and which part allowed modification without loss of activity. In short, the lactam was arylated, followed by the condensation of the keto function of the lactam with a functionalized arylamine, and upon treatment of these compounds with a strong base, the formed anion condenses with the methyl ester, forming the quinolone that is the basic skeleton of blebbistatin. The final step for the introduction of the chiral alcohol function can be performed by an enantioselective epoxidation via the corresponding enantiopure Davis oxaziridine, followed by ring opening. Better results could be obtained by combining the 2 final steps in a one-pot strategy. Using similar strategies, it was possible to make ring A analogues with extended aromatic ring systems, as well as with aliphatic substituents. On the next slide, the produced D-ring analogues are presented. As can be seen, a wide variety of D-ring analogues have been prepared, and the physicochemical properties, water solubility, and the light stability of the compounds could be tested. In summary, the ring A and ring C analogues did not result in any improved characteristics. Lots of work has been performed, but without any success. However, among the D-ring analogues, the hydroxy and amino substituted derivatives showed really improved characteristics, with a water solubility that was 30 times higher than the parent blebbistatin, with the conservation of the cell permeability and with no fluorescence interference, as can be seen on the picture of a fluorescence microscope. The top picture shows the view of blebbistatin in cells with lots of fluorescence and which is not desired, while the bottom picture shows the view of the amino analog with no interfering fluorescence. In summary, in all our projects, we try to use organic synthesis to create novel chemistry and chemical diversity in order to innovate in the medicinal sector. I want to thank all present and past collaborators of the SynBioC group, and I would like to thank you for your kind attention.